Hi guys, we're about to start the next section of immunology. This is an immunodiagnostics section, and so I thought I would maybe tap into what's going on right now with COVID-19 and kind of use that as an example and talk about how diagnostics might be involved. I've got a couple of colleagues here to help me with this today, and so I'll let them introduce themselves.
Hi guys, I'm Michael, second year veterinary student.
I'm Minas Mkhitaryan, third year vet student.
So you guys are going to help me out talk about coronaviruses and then COVID-19, so let's go to it, okay?
Let's do it.
So first, a little background, guys. And so the first thing is I want to remind you guys that COVID-19 is a disease, and it's caused by coronavirus, and the specific coronavirus is SARS-CoV-2, okay? So a few things first about coronaviruses. So there's lots of them, and they're an envelope virus, and they're an RNA virus, and they have a single, pretty long strand of RNA. And it's a non-segmented genome, so it means that there's no recombination with different segments of the genome, but it's one big piece. Okay. Now, coronaviruses, you guys heard of them in vet school. You know, there's a lot of them, and in animals they mostly cause GI diseases, but they've been around in people for a long time too. Where they cause mostly upper respiratory signs, and so, you know, prior to kind of SARS, the beginning of the 2000s, we just thought them as causing common colds, and like 15 to 30% of human colds are caused by coronaviruses, so they're kind of floating around. And mostly they're just mild diseases. It's more severe in neonates and in old people and people who are immunosuppressed or have some underlying sickness. But they're around, they're there, and mostly they've not been a problem in the past. Okay, and so that's kind of what coronaviruses are, just to remind you. And so this one is SARS-CoV-2. That's what it's called. But there's a couple different kinds. And right here is just a kind of an example of a kind of a prototypic coronavirus. So you can see in the middle here, point out to you guys, that's the RNA genome. It's one long strand of RNA. It's got an envelope, and then it's got these structural proteins. There's like 4 kind of main structural proteins. Some are exposed on the surface and some are buried. It doesn't matter what you call it, what the names of them are, but that's kind of how they're all put together. So when animals infected, they use these, they bind to a receptor on the cell. What receptor they bind to depends on the virus and the species and things like that.
Yeah.
Like animals are GI, these others are respiratory. COVID-2 binds deep in the lungs, that's what causes such severe problems.
I see.
So when a virus infects, there's something called an incubation period. So do you guys remember what an incubation period is?
Yeah, so incubation period is the time between when you're exposed to the pathogen and when you start to show symptoms.
Okay, so when clinical signs appear.
Yeah.
What do you know about the incubation period for COVID-19?
I heard it's pretty short. I heard for the most part it's like 2 to 7 days.
Yeah, that's right.
Time period.
Kind of in that period. Yeah, so that's perfect, guys. The incubation period is that time between when some animal or a person encounters a pathogen and when they start showing clinical signs. And there's a recent study that showed that at least in the Wuhan province, that SARS-CoV-2, the median incubation period was about 5 days. And the range is somewhere between about 2 and 12 days. Like 97% of people who got sick showed signs in less than 12 days. So it's pretty short.
Yeah.
And that kind of comes into play with the quarantine periods too. They're 14
days.
Yeah, right. You probably heard that.
Yeah.
Okay, so created a couple of figures here to kind of illustrate a couple of points. And here's again is this coronavirus, kind of prototypic coronavirus. Let's say that's a common human coronavirus. And I took that and created a new variant. Okay, so a different virus now. As you can see, what I've done is I've changed some genes here in this yellow here. So this coronavirus has some different genes in its genome.
Yeah.
You see anything different else that might be reflected in that figure?
Yeah, I noticed that there's kind of a novel antigen here. These yellow boxes here on the outside of the virus.
Yeah. So in this case, that genetic difference is been translated into some protein differences, and so those are expressed on the surface. So that means that the spike proteins are a little different than this virus compared to this other virus here. Right?
Yeah.
Okay, just to take that one step further, in this figure here I created 2 more coronaviruses. So now we have 4 human coronaviruses. Do you guys see any can you see the differences that I created in these 2 new ones?
Kind of, yeah. On this one here, as we also see the difference in the RNA sequence
Okay.
and how that change part of the proteins embedded in the membrane all around here.
Okay.
And also, I see on this one, there's some change in the sequence
Okay.
and change in the proteins that's inside the membrane,
Okay.
which will probably, I don't know, probably be, there's variation in detection in that way,
Yeah.
and kind of how they interact with the body and the antibodies.
What do you know what he means? That's when he says about the fact that there might be differences or not on the outside.
Are you referring to the fact that these are inside? And
Right.
yeah, I think what he means by that is if you had the body producing antibodies to fight against this virus, they might not be able to see these protein differences that are within the virus. But that doesn't mean that the immune system through T cells or otherwise might not be able to pick up on those.
Yeah, they might have unique T cell epitopes that when they get processed, they become part of the immune system, but they're not going to be available to antibodies on the outside.
Yeah.
So that's kind of our 4 different coronaviruses. So let's maybe say 3 of these cause colds and one of them causes COVID-19, something like that. All right. So there's been a lot in the news about the diagnostic test for COVID-19. So what have you heard about this diagnostic test?
I heard that they use swabs.
Okay.
So sample, they swab the nose or the mouth, and there's different ways they test for it. I know the United States tests in a different manner than Australia or the UK. And as you could see on the picture on the top left, there's drive-through testing facilities that have been set up.
Mm-hmm. Anything else about the testing?
Yeah, I mean, one thing we know is that they haven't found like the ideal test yet. There's not enough of the right kind of test, so they're actively searching for something better.
Right. The big thing really is that in the U.S. anyway, there's not enough of them. Right.
Mm-hmm.
We didn't have enough in hand, and like Michael said, the test we're using a slightly different test, but the bottom line is there's not enough to go around. So a lot of people who want to be tested haven't been tested or can't be tested. All right. So here's a question for you guys, for the students in the first year class. What I want you to do is to go to the top hat. So remember, for this immunodiagnostics, and I want you to take just a minute to answer a multiple choice question there, and that is, what kind of test is the SARS-CoV-2 test, the current test that's out there right now? So just take a minute, go look at the options and see what kind of test it is. No points involved. There's nothing lost here. And so pause this video, hit pause right now or in a couple seconds, and then go do that and then come back when you're done, okay? And then unpause it and go forward. So go answer that top hat question. And meanwhile, we'll sit here and drink coffee or something. All right, go for it, guys. Think they'll do it?
Nah.
The smart ones will.
I hope so. Hopefully they do it, because if they do, that makes them think about it. They'll remember it longer if they have to think about it a little bit. So let's hope they do that. All right, let's assume that they're back. We'll move on and talk about it. So I'm going to ask you guys, what is the current SARS-CoV diagnostic test? So the thing I asked the students was, is it a serologic test, an antibody test, an antigen detection test, a genetic test, or some combination of those things? So what kind of test is it?
It's a genetic test.
Okay.
Yeah. Using a method called PCR, polymerase chain reaction.
Okay.
They essentially, they look at the virus genetic composition and try to identify it through its genome.
Okay. I got a little figure here. Let's see if you can explain it. Can you guys explain how the test works a little bit? Just looking at this simple figure that I stole from the internet and modified a little bit.
Yeah, so as you mentioned before, it's an RNA virus.
Okay.
So
you can use the pointer if you want.
Yeah, they take the RNA and they use reverse transcriptase, and they get a copy DNA.
Okay.
And with the PCR method and amplifying specific genes that's specific to CoV-2, and then they test for it in that manner.
Okay.
I don't know, where's my mouse? Oh, it's right here.
There you go.
So it has to be specific. They find specific nucleotides within that sequence that is especially specific to SARS-CoV-2.
Right. And that's one of the reasons the CDC US test is different than the other tests. Are you looking at different genes than everyone else in the world is what I'm looking for?
Oh,
yeah.
Different sequences within
Right.
the virus.
Right. So it's not an immunodiagnostic test at all.
So why are we doing this, Steve?
Well, we'll get to that. Be patient. Okay, so there's a couple questions for these guys to think about, for you guys to think about here. And again, I want you to go to Top Hat and answer these two questions. The first one is a multiple choice question, and the second is a discussion question. So first of all, I'm going to ask these guys this in a minute. Why might an antibody detection test be useful? What might be an advantage or something specific about an antibody test versus a genetic test that we just described? Okay, and the next question for you guys to consider is, okay, based on what you just learned about coronaviruses, including SARS-CoV-2, what would be the particular challenges in developing an antibody detection test that you could be used to detect SARS-CoV-2 infection? All right, so go to Top Hat. So first, pause this. Go to Top Hat. Take a few minutes and think about these. Answer number 2, the multiple choice question, and give it a few minutes and answer at least one possible reason for question number 3 in the discussion question that's posed there. Okay? And once you've done that, come back to this video, unpause it, and we'll talk more about it then. But please go do that. I think you'll remember and learn a lot more if you do that first, and just give it a shot. And no penalties if you don't get it right. All right, go. Any chance?
I think a little more will probably answer it this time. This is interesting stuff.
You had a compelling argument too. So.
Yeah,
compelling argument. You guys
There's no points to lose.
Time. All right, well, we'll see. Hopefully they'll come back now. You guys back? All right, let's try this again. So we're just going to have a bit of discussion now about these next couple of questions, and this is where we're going to set up our next section of immunology. So here's our 2 questions. So first, why would an antibody detection test be useful in compared to genetic tests? So who wants to answer this one?
I can get that.
Okay.
I think one reason that it's particularly useful is because in order to pick up on viral genome, the patient has to be actively infected.
Okay.
And if they're not, let's say they've been infected and they've recovered.
Okay.
You're going to test negative.
Okay.
And if you're interested in learning about who was infected, kind of retrospectively, an antibody test would be the most helpful in that scenario.
Okay. What would be the downsides? What would be the challenges of doing that, Michael?
So as we talked about, the incubation period, how is this very short?
Okay.
So you'd have to test. The test has to probably be an IgM test, and
Okay.
early on in the infection.
Okay.
And what would be another reason? I feel like also with the figures that we mentioned before, how there's similarities between the coronaviruses.
Okay.
So the test, if you're going to use an antibody test, has to be specific to COVID-2,
Right.
and not cross-reactive in that way.
You wouldn't want to be able to detect. You wouldn't want to be detecting antibodies from people who had coronavirus colds.
Yeah, different coronavirus.
I think they had SARS.
So it has to be a very specific test too.
Okay.
Yeah.
So Minas, you mentioned the idea of kind of retrospective. Why would you want to know whether people had previously been infected and recovered from SARS-CoV-2?
I think it might be helpful in understanding like the epidemiology of the disease.
Okay.
Because I don't know, let's say this comes back next year. I think learning about how this thing spreads and how people are affected by it would be useful.
Yeah. I mean, one of the big questions right now, I think, is how many people are having really mild signs or no signs but are infecting other people.
Mm-hmm.
We know people who are sick or they shed lots of virus, and they're the biggest risks to other people. But we don't know yet, anyway, what else might be going on.
Mm.
So it's going to be hard to manage and control this disease if we don't understand how it's transmitted. So, good questions. Just to kind of illustrate the stuff that Michael was talking about. Michael was mentioning that, if you have antibody detected against some like these spike proteins here, if it detected also against this one over here that cross-reacted with a coronavirus that caused common colds, it would be positive as well.
Yep.
You'd want to have it against this epitope that's an epitope that's specific for the virus you're looking for. And so you have to know a lot about the genome, which we do, and so probably that could happen.
So maybe I wanted to clarify. So
Yeah.
you're saying if we have a test that detects for this epitope here, so there will be cross-reactivity. So we have to test
Right.
for this protein that's on the envelope
Right.
to be specifically for the COVID-2.
Right, exactly. And even then, if it bound to this part of the protein that's the same, so the green, that's still going
That's
to
right.
cross-react. So it's got to be epitope specific, probably bind something that's really unique to one virus and not the other.
Okay.
So it's not the whole virus the whole protein may not be different, but parts of it will be. Maybe that's what makes the virus more virulent or allows it to bind to a different receptor. Yeah, so it's going to be a pretty key question. I found this on the internet. There actually is, at least experimentally, a test out here, and Minas mentioned the IgM versus IgG, and you can kind of differentiate them. And this is experimental. It's not FDA approved. Because of the emergency situation, these tests are kind of being rushed out there. I think it's based on some of the genetic sequences they know are unique to SARS-CoV-2. But if you look at the fine print, even now it says positive results may be due to past or persistent infection and the non-SARS-CoV-2 coronaviruses. So it probably does not cross-react, but we don't know that yet. It hasn't been validated. So that's some of the issues we're talking about, right?
So this might be like a CYA thing in the meantime until it's validated.
Right, until it gets validated. And people working like crazy on these sort of things. They're also working on antigen detection systems. Why might that be useful? So what would be the advantage of an antigen detection system over a genetic test?
Faster.
Yeah, faster.
Yeah, you could do like the SNAP test we have in veterinary clinics for parvo or FIV or whatever.
Theoretically, you could have a result in 30 minutes, right? Or in a short amount of time if you had an antigen detection. But again, same problem, right? It's going to have to detect an antigen that's unique to the COVID-2 and not to other coronaviruses. This just shows the thing that you were talking about, Michael.
Yeah.
This is an immune response to a pathogen. You want to point out that thing about the IgM thing?
Yeah, so we mentioned the incubation period is on average 5 days, so somewhere in between here. And then your body responds with IgMs.
Right.
So you would want to the test would have to be active here within this period, or be tested within this period, because if it's probably not going to catch the IgMs later on in the infection.
Yeah, and another thing is that the person might be shedding before you pick up on IgM,
Right.
Especially if they're elderly.
Right.
Their IgM response is going to be more delayed.
Right.
Yeah.
You can see you can use it later on. You can look for IgGs in particular. It'd be IgG high and IgM low, and that would tell you they'd been infected sometime in the past.
If it's acute or chronic.
Yeah, it'll let you know how much the virus has been circulating, causing problems that we didn't even know about. All right, guys. We're going to talk more about antibody-based tests in this section. We're going to talk about some diseases where IgM tests are actually quite useful. They have a little longer incubation period than COVID-19. That helps a little bit. And we'll go from there. So, I hope this wasn't totally lame, you guys. And I want to thank you guys for helping me out with this. I think it'll be a good lead-in to the next section. And class of 2023, let me know what you think. Otherwise, thank you guys.
Thank you.
See you soon.